We often think of disease as an external invader, a bacteria or virus breaching our defenses. Cancer is an internal rebellion. It occurs when one of our own cells goes rogue, rewriting its own genetic instructions to divide and conquer. Because these cells share our own genetic code, they present a unique challenge. The immune system must distinguish healthy tissue from a localized rebellion without attacking the body itself. The answer lies in the fact that rapid, chaotic mutation is sloppy work. As a tumor warps its internal machinery, it accidentally leaves behind distinct biological signatures, a microscopic fingerprint of its betrayal. Survival depends on the body's ability to identify and interpret these signatures before the rogue cell has a chance to multiply. In the rush to mutate, tumors reactivate ancient locked DNA segments from embryonic development. Biologists call these oncofetal antigens. Reactivating them places a massive biological target on the tumor's surface, acting like a flashing beacon alerting the immune system. This technical data illustrates how we turn that error against the cancer. Doctors track these embryonic markers, specifically AFP and CEA, to diagnose and monitor liver and colon tumors long before they are visible on a standard scan. These genetic leftovers provide the immune system with the precise coordinates required for a targeted strike. Once a target is identified, the body deploys its elite forces, cytotoxic T cells. These are the immune system's precision snipers. They do not shoot blindly. These cells perform a high-resolution scan of the target's MHC class I molecules, a biochemical ID tag, to verify the cell's traitor status before engaging. This diagram outlines the lethal mechanism. Once the match is confirmed, the T cell triggers a biochemical sequence that forces the cancer cell to dismantle itself from the inside out. If a tumor attempts to hide from the snipers, the body sends in natural killer, or NK cells. As shown here, they bypass the precise ID scan and punch holes straight through the membrane of anything acting suspiciously. Under normal conditions, these forces win easily. Your body likely identifies and clears microcancers every day without you ever knowing. If we possess an army of assassins, why is cancer still a leading cause of death? The answer lies in the tumor's ability to evolve sophisticated stealth technology. The first evasion tactic is cloaking. The tumor pulls identifying MHC proteins back inside its membrane, leaving T cells scanning empty space. Next, it deploys decoys. The tumor sheds a cloud of loose antigens, tricking antibodies into binding with harmless debris instead of the cancer. It also constructs physical bunkers. By hijacking the clotting response, the tumor weaves a dense fibrin cocoon around its perimeter, creating an impenetrable wall. Research documented here confirms that tumors even engage in chemical warfare. They secrete compounds like TGF-alpha to paralyze any macrophages or lymphocytes that get too close. A lethal tumor is an active combatant in an information and chemical war that natural biology struggles to win. When biology hits its limit, human science takes over. We are now rewriting the rules of engagement with immunotherapy. Instead of poisoning the whole body, we are engineering super soldiers known as lymphokine-activated killer cells, or LAK cells. This diagram outlines the process. We extract a patient's natural killer cells and saturate them with the signaling protein IL-2 in a lab. This bypasses their natural limits, creating hyper-aggressive LAK cells that we then inject back into the patient. We also use conjugate antibodies, specialized structures that act as biological handcuffs, physically tethering a T cell sniper directly to its stealthy target. Rather than functioning as a generic toxin, these treatments work by removing the biological brakes the tumor uses to evade detection. Perhaps the most cinematic tactic in modern oncology is the biological false flag operation. If a tumor is invisible to the body's radar, scientists inject flu virus antigens directly into the center of the mass. The immune system rushes to the site, convinced it's fighting a massive infection. It carpet bombs the area, destroying the cancer as collateral damage. Ultimately, curing cancer depends on winning this intelligence war. By exposing exactly how the traitor hides, we can deploy a biological software update that ensures our internal army wins the final battle.